Hi micro folks, we're back March 13th, 2020. We're calling these the coronavirus tapes. So these are some lab videos. Hopefully you'll be able to see how we would have done these techniques if we don't get to do them in person. And so this is going to be a short video on how to inoculate our lab manual chapter 16 metabolic tests. And specifically, we're going to be looking to see if our different microbes can ferment different sugars, and we'll be using what's called PR, sugar or carbohydrate fermentation medium. And then we'll also be testing our microbes' ability to grow anaerobically or aerobically using a medium called Thioglycollate. I won't video record all of the inoculations because they get pretty repetitive, but we'll try to give you an introduction to the medium, and then we'll have a follow-up PowerPoint that will give you more details. And certainly, folks, reading Chapter 16, all the information will be in Chapter 16, the lab manual. In the spring 2020, Chapter 16 starts on page 173. And a table showing the organisms that we would have inoculated and which media we would have used. A table is on page 178. So folks, we'll first start out talking about PR, sugar or carbohydrate fermentation medium. So here's a tube of our PR, sugar fermentation medium. The PR sugar fermentation media has a single sugar, and the medium will be named after the sugar. So for example, this is PR glucose, telling me that there's only glucose in this medium. There's also proteins and amino acids as alternate terminal electron acceptors. In addition, there is an inverted glass tube that's open at the bottom, closed at the end, and that's called the Durham tube, D-U-R-H-A-M. And the function of the Durham tube is to collect gases produced during fermentation. And those gases would be carbon dioxide or molecular hydrogen or both. And after incubation, we would see the gases collected as a little bubble in the end of the Durham tube. The pH indicator in our PR means phenol red, the pH indicator. Phenol red will turn yellow at acidic pH and will turn a darker red at alkaline pH, kind of a fuchsia color. Now, the basis for this test is that if our microbes can ferment the sugar, they'll produce acids. The pH will drop, and under acidic pH, the phenol red will turn yellow. So if the medium is yellow after incubation, that means your microbe could ferment the sugar. If the medium is red or fuchsia color, that means your microbe couldn't ferment the sugar. Instead, it tore apart the amino acids, releasing the amino group as ammonia, which we know is a weak base. The pH increases, and the phenol red turns that deep red or fuchsia color. Okay, so I'll show you just quickly how we would use aseptic technique to inoculate 3 PR sugars. We're going to use PR glucose, PR lactose, and PR sucrose. And what I'll do is just use our good friend E. coli. E. coli is so adaptable, and E. coli, we know for sure that it can ferment glucose. Okay, in our metabolism discussions, often E. coli is kind of our model workhorse. So we know that E. coli can ferment glucose. It uses a specific fermentation pathway called the mixed acid fermentation pathway, but we'll come back to that again. But in doing this test, now we'll get to see if, in addition to glucose, if E. coli can also ferment lactose and sucrose. Now, glucose, if you're going to ferment a sugar, it's going to be glucose. It's a monosaccharide. If you're going to ferment lactose, you need an additional enzyme. You need the beta-galactosidase, the enzyme that will hydrolyze a glycosidic bond between glucose and galactose in lactose. And if you're going to ferment sucrose, you'll need an additional enzyme called sucrase, which will hydrolyze the glycosidic bond between glucose and fructose in sucrose. Okay, so you guys, I've I've sterilized my loop. This is my pure culture of E. coli. I'm going to just touch the agar to make sure that it is cool enough. I gather some E. coli, and this is going to get repetitive, you guys. We're just going to warm the cap. We're going to warm the lips, and then I'm going to insert my loop. And again, folks, all I really need to do is just touch the loop to the liquid medium, and I should have lots of E. coli falling off, but I'm kind of a bad habit. I like to swish it around just a little bit. Okay, and I'm going to withdraw, heat the lips, heat the cap. Okay, and I want to make sure my cap is loose, not really loose, but loose enough so the gases can vent, right? Otherwise, the gases could cause the tube to explode like a hand grenade. And then I'm going to sterilize my loop. And then, folks, I would just repeat that same procedure to inoculate my PR lactose with my E. coli, to inoculate my PR sucrose with my E. coli. And then there will be other microbes that will inoculate, so we can compare fermentation patterns between E. coli and the other microbes. So fermentation patterns, you can think of them almost as a metabolic fingerprint. So different microbes will be able to carry out different metabolic processes, different fermentation, fermentation of different sugars. So when we come in and look at our results, we'll be able to see, we could actually start identifying our microbes based on which sugars they can and cannot ferment. The last test we're going to show you here is a test for the ability of our microbes to grow aerobically or anaerobically. So let me just go back in here and re-sterilize. And again, folks, I'll just show you how to inoculate the thioglycollate medium. The thioglycollate medium, thioglycollate reduces molecular oxygen. So with thioglycollate medium, I should move this up here so you can see. Okay, we would presume that up at the top, at the air broth interface, there would be a little oxygen present. And indeed, folks, we have an oxidation reduction indicator present, and it's green when oxygen is present. But you can see down below here there's no green. That's telling us this is an anaerobic compartment. Okay, and again, the thioglycolate has reduced the molecular oxygen, so we have a nice anaerobic compartment here. So I'm going to inoculate my E. coli, and this time I won't try to swish my loop back and forth, because I do not want to introduce air into my thioglycolate media. So we're really careful. We're going to inoculate straight down and straight back up, and we want to be really careful not to shake the tubes, right? Because we want to maintain that anaerobic zone there. So again, just really quick, we'll sterilize my loop. I'll open my pure culture plate of E. coli, touch the agar to make sure my loop's not too hot. And then all I have to do is just barely touch where the E. coli is growing, just get a little bit on my loop. And then this is getting repetitive, right? Warm the cap, warm the lips. And again, folks, I'm going to go straight down. I'm not going to swish my loop, and I'm going to come straight back up. Warm the lips, replace the cap. Make sure it's not too tight. Warm the cap and replace my tube. And always, you guys remember, always, we're going to sterilize our loop before we're finished. Okay, folks, so what I think I'll do here, we'll keep this short. I did want to show you, folks, how we are going to incubate our tubes. We are very sophisticated. We just bring cans from home, clean cans from home. We're going to put a paper towel in the bottom of the can, and then we're going to spray the paper towel with disinfectant. And the reason for this is, we'll see in some of our metabolic tests, the microbes are going to make so much gas that they'll, especially in our agar slants, they'll make so much gas that the slant will explode, and part of the slant will get pushed up against the cap, and there's so much pressure, so much gas pressure, that sometimes the agar and the microbes squeeze out under the cap and onto the sides of the tube. So we want to make sure that we have a disinfectant soap paper towel in here to help to inactivate any of the microbes get pushed out. And also, before we handle the tubes, we're going to spray them with disinfectant again. again, because there might be microbial contamination on the outside of the tubes. And then the way we incubate, folks, we just put all of our tubes into these cans, double checking to make sure that the caps, I tighten them and then just barely loosen to vent them. We're going to put all of our tubes in here, and then we're going to incubate and then read the results in 48 hours. So we'll come in this weekend to read the results on our PR sugar fermentation tubes and our Thioglycollate tubes. Okay. We'll do one last video, folks, and I think that might be it. And that will be on Chapter 17, Metabolic Test Part 2. We're going to look at the IMVIC tests. We're going to look at TSI, triple sugar iron slants, and we're going to look at the catalase test. And then we'll be probably finished with our lab videos.